My name is Maureen Gannon. I'm an associate professor in the Department of Medicine in the Division of Diabetes, Endocrinology, and Metabolism. My laboratory studies factors that regulate the formation of insulin-producing cells in the developing embryonic pancreas, and also factors that regulate the expansion of those cells in response to different physiological stressors, such as pregnancy and weight gain. The insulin-producing beta cells are the only cells in the body that make insulin, which is the hormone required to maintain normal blood sugar. What you're looking at here is a section of a mouse pancreas, although if you were to look at a tissue section from human or any other mammal, it would look pretty much the same. The insulin-producing beta cells are labeled here in green, and the glucagon-producing alpha cells are labeled in red. These two cell types together help to maintain normal blood sugar levels, with insulin causing a decrease in blood sugar and glucagon causing an increase in blood sugar, for example, in response to fasting. The blue that you see here marks the DNA in the nuclei of all the different cells in the pancreas. Prior to the discovery of insulin in 1921 by Banting and Best in Canada, it was unknown which hormones regulated blood sugar levels. When they discovered that insulin was the pancreatic hormone that regulated blood sugar, it was really a life-changing discovery. Prior to that discovery, a diagnosis of type 1 diabetes, in which the immune system destroys the pancreatic beta cells, this was actually a lethal diagnosis. On the left here, you can see a child, JL, at 3 years of age, who has type 1 diabetes. And this is prior to his treatment with insulin. And I think you can appreciate that once this child was treated with even a very crude preparation in the early 1920s of insulin, you can see that he has now gained weight and is much healthier, even though he has type 1 diabetes. So treatment with insulin is absolutely required for type 1 diabetic patients. People who have type 2 diabetes, on the other hand, which is the vast majority of people with diabetes, still have pancreatic beta cells. But these insulin-producing beta cells are insufficient to maintain normal glucose levels in the face of obesity and increased weight gain and insulin resistance. So both type 1 and type 2 diabetes patients have an insufficient number of insulin-producing beta cells to maintain normal blood sugar. In this cartoon, you can see the pancreas in yellow in the upper left-hand corner, and your pancreas is located right here in your abdomen, below your stomach, near your small intestine. The insulin-producing beta cells in the pancreas, as I mentioned, are the only insulin-producing cells in the body. So if they're destroyed by the immune system in type 1 diabetes, or they're not functioning properly in the case of type 2 diabetes, there's no other cell that can replace them. In this cartoon, I show you how insulin works in the body. In the second circle in the middle, you can see a blow-up of an insulin-producing beta cell. These cells secrete insulin directly into the bloodstream in response to glucose elevation in the blood, which usually happens after you eat a meal. Once the insulin is circulating through the blood, it goes to target tissues throughout the body, such as the muscle, the liver, and the adipose tissue, or the fat tissue. And there the insulin interacts with proteins called receptors on the cell that cause the uptake of insulin into the cell and stimulate then the uptake of glucose from the bloodstream. In the absence of sufficient insulin, the glucose would remain in the bloodstream and would not be taken up by the cells to be stored for energy or to be used for future purposes in the storage form of glycogen or fat in the case of the adipose tissue. So you could imagine then that if we could restore the beta cells or the insulin-producing cells in an individual's pancreas who has type 1 diabetes, or increase the number of functional insulin-producing cells in an individual who has type 2 diabetes, they would no longer have to rely on insulin injections in the case of type 1 diabetes, or on oral medication in the case of type 2 diabetes. And this is something that my lab is very interested in doing. We want to identify ways that we can actually generate a cell-based therapy for the treatment of type 1 or type 2 diabetes, where these individuals would either have transplanted functional insulin-producing beta cells or be able to generate in their own pancreas new functioning insulin-producing beta cells, and they would no longer have to rely on medication. So here I've outlined 4 different strategies that my lab and other labs around the world are taking in order to generate replacement beta cells, insulin-producing cells, for patients with diabetes. And we're going to go through these each in turn. But just to give you an overview, we're going to talk about number 1, where we can use beta cells from cadaver donor islets and expand those in culture and then hopefully transplant those into patients. In number 2, we're going to talk about the potential for cells within an adult individual's own pancreas to generate new beta cells or insulin-producing cells. In number 3, we're going to talk about strategies that people are utilizing to generate beta cells from embryonic stem cells or from adult pluripotent stem cells. And then in number 4, we're going to talk about strategies that people are using to try and convert closely related cells like acinar cells in the pancreas that make digestive enzymes and convert those cells into beta cells in a person's own body. So let's go through these in turn. The first strategy is to use cadaver donor beta cells and to expand them in culture and use them to replace beta cells in a patient with diabetes. And is this even possible? This is something my lab has been working on quite extensively. We get isolated islets from either mice or from human cadaver donors through a national network, and we can treat them in culture with different factors and combinations of factors. And here's an example of one of those. We've been studying this factor called connective tissue growth factor for a number of years now. It's CTGF for short. And what you're looking at on the left-hand side, you see in green, you see the insulin-producing cells. Again, blue is the DNA and the nuclei. And any nucleus that's labeled in pink is undergoing proliferation or expansion to increase the number of insulin-producing cells. When we add increasing concentrations of CTGF to islets in a culture dish, as you can see on the right-hand side in the graph, we get an increased number of the beta cells that are proliferating and therefore an increase in the number of insulin-producing cells. So this shows us that at least in a culture dish, CTGF can induce an increase in the number of insulin-producing cells. But can it do this in vivo? We have some evidence that it can. We've been using a mouse model where we can, using a genetic trick, we can kill 50% of the insulin-producing beta cells in the pancreas. So if you compare in the upper left-hand corner, a normal islet, again, insulin is in green, with an islet from a mouse in which we've destroyed 50% of the insulin-producing cells, you can see that we have a dramatic decrease in the number of the green cells. When we overexpress or induce expression of CTGF in these remaining insulin-producing cells, we get an increase in beta cell replication or beta cell division. And that's shown in column number 4, where those mice have been treated with CTGF, and we see an increase in the number of beta cells or insulin cells that are rapidly dividing. This translates into a regeneration of beta cell mass. As you can see again in column number 4, there's an increase in the number of beta cells when we treat with CTGF. So we have evidence that this factor can induce regeneration of insulin cells in the pancreas of a mouse. So moving on to strategy number 2, we and others would like to identify methods where we could induce the formation of new insulin-producing cells in an individual's own pancreas. This is very controversial in the field right now. Some people have gotten it to work in mice and some haven't, and so I'm not going to really talk about this very much except to say that there's evidence in humans that this can actually happen. What you're looking at here is a tissue section from a cadaver, a human patient, where we've stained with insulin in brown, and then the rest of the tissue is just labeled in blue. And what you're looking at here are insulin-producing cells that are very closely associated with these tubular structures, which are called pancreatic ducts. In the developing embryo, this is where the insulin-producing cells come from. In an adult, you would not normally see this. However, this is a pancreas from a patient who had type 1 diabetes for a very long time. And what I think you can see is that even though this person's immune system is attacking their beta cells on an ongoing basis, they are still attempting to generate new insulin-producing cells from their pancreatic ducts. And you can see the brown cells very close to the ductal epithelium. This is also observed in pregnancy and obesity. So we think this is strong evidence that there are certain circumstances and certain signals that can cause the production of new insulin cells from pancreatic ducts, even in an adult, and that this area is really worth pursuing. The next strategy we're going to talk about is using stem cells, either embryonic stem cells or adult pluripotent stem cells, and using information from developmental biology to generate insulin-producing cells. And what you're looking at here with the gray arrows is a pathway that a stem cell in an embryo would normally go down. So they first become endoderm, then pancreas, and then beta cells. And there's certain signals, growth factors, transcription factors that we know from developmental biology are absolutely essential for a stem cell in an embryo to eventually become an insulin-producing beta cell. And with that knowledge then, we can take these stem cells, put them in culture, activate these pathways in a particular order, and hopefully generate insulin-producing cells in a culture dish. And this has been very successful. And what you're looking at here is a cartoon describing a pathway of differentiation from a stem cell all the way on the left to a fully differentiated insulin-producing cell all the way on the right. And there are steps that we have to go through here, 5 steps delineated in this particular protocol. And people have shown that at the end of this protocol, they can get about 13% of the cells in the dish that are producing insulin. It's not very efficient, but it is happening. Others have shown that if they take these cells at one stage earlier than a fully differentiated insulin-producing cell and transplant those cells into a mouse, that these cells very efficiently differentiate into insulin-producing cells and can reduce blood sugar in a mouse with diabetes. So these protocols from embryonic stem cells or induced pluripotent stem cells have been highly successful. And in fact, there are clinical trials ongoing right now sponsored by the Juvenile Diabetes Foundation where these cells are being implanted in encapsulation devices kind of about the size of a tea bag into patients who do not have diabetes right now, but they're just being tested for safety and efficacy. And hopefully we'll be moving on into patients with type 1 diabetes very soon. Finally, the 4th strategy that people are using is to differentiate other cells in the body into insulin-producing cells. So there are cells in your pancreas called exocrine cells that make digestive enzymes, hepatocytes in your liver, and hormone-producing cells in your intestine that if you just add a few crucial genes that are important for the differentiation of an insulin-producing cell can actually convert these closely related cells into insulin-producing cells. What you're looking at here on the right is an electron micrograph of an insulin-producing cell from a mouse pancreas. The insulin is packaged into granules. You can see these circular-like structures with a dark center. The dark center in the middle are the insulin crystals, and they're going to be secreted right into that adjacent capillary. And people have shown that just by introducing 3 transcription factors into an exocrine cell in the pancreas, you can convert them into a cell that looks exactly like a beta cell would normally look. And it produces insulin and lowers blood sugar. So I'd just like to summarize by emphasizing again, there are lots of different strategies that people are taking. Many of them involve using information that we've gained from developmental biology of the pancreas and trying to use this information to coax either replication of insulin-producing cells and generate more of those cells, or coax other cells in the body to produce new insulin-producing cells. And that we're very hopeful in the field that all of these strategies together are going to really be successful in generating replacement insulin-producing cells for individuals who have diabetes.